Jason.
Oh, okay. How are you? So fortunate to have these wonderful people with you. So make best use of them. I mean, you can't make me, right?
Nice one.
Please.
Distinguished guest speakers, dear participants in person and online, and comstech team, ladies and gentlemen, Assalamu alaikum. I welcome you all to today's event, Comstech International Seminar on Advances in Neglected Tropical Diseases. We are joined today by renowned international experts, Professor Dr. Paul Denny, who is a professor at the Department of Biosciences and at the Department of Chemistry at Durham University in UK. His research interests include cell biology, lipid biochemistry, molecular biology, and parasitology. And we are also joined today by Professor Dr. M.K. Paul, who is a professor in the Department of Biochemistry, Biosciences and Chemistry. And his research group works mainly on protein crystallography and DNA binding proteins. Thank you very much, sir, for gracing us with your presence today. So I would like to formally begin the session with the recitation of the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان وأقيموا الوزن بالقسط ولا تخسروا الميزان صدق الله العظيم.
Next, I would request His Excellency Professor Dr. Muhammad Iqbal Chaudhary, who is the Coordinator General of Comstech, for his welcome address, please.
Mic me? You can hear me? Okay. So, Assalamu alaikum and good morning, girls and boys, a lot of young people. I think this youthful participant is something which really they give me a lot of hope because the eagerness to learn is so extremely important. And for that, we have today with us two very prominent speakers and also very dear friends who are working with us. since last 5 years. We first met in 2018 when we became the part of Pakistan, officially became part of a large network of 6 countries, focusing on leishmaniasis and trypsinoma. These 2 diseases were the focus, and these are neglected tropical diseases. Along with us, of course, this was led by a Durham team, Paul, MK, Patrick, Steve, and others. And there were countries like Uruguay, Argentina, Brazil, India, and Pakistan. Six of these countries became part of a very large network for a cause and a mission, and that was working on neglected tropical areas. This is a continuity of the same collaboration which we have established in 2018, and then friendship, which came along with this. And I think this is really, along with the good quality of science which network has done, we were also able to develop productive collaboration and friendship beyond the project cycle. So project ended last year. Summer is sitting here, she's coordinating the efforts. She was coordinating the network activities in Pakistan also. But I'll tell you what we were able to do, along with various projects, instrument grants and others, in terms of capacity building. So in 2019, we had this hands-on training course, an extremely important event in which our colleagues actually have visited hospitals where they have actually seen people with cutaneous leishmaniasis, a very common disease in Pakistan. And as a result of it, there were a lot of public outreach because, our dear friend Max have then developed an animation which you'll be able to see on on YouTube if you wish to, of how these neglected tropical diseases affect families, communities, and countries. And if I give you statistics, you would perhaps be surprised that Pakistan is among the top 10 countries in terms of neglected tropical disease burden. And the number of neglected tropical diseases are as much as the total count. So hardly anything which is not there, from literally roundworm to dengue and Zika and leprosy and you just name it. And then on top of it, increasing burden of cutaneous leishmaniasis, which is a very important disease. So we had this event in 2019 in Karachi. Then we organized a training course in Kenya, Nairobi, for the East Africa. We are planning to have another very important training course in Ethiopia. This is planned in March, hopefully, and February, depending on the availability of the experts. And then we also had, I think, during COVID-19, where mobility was not possible, we continued to work, and comstech and NTD had a series of lectures, online lectures given by top experts and members of the network too. We are also planning to, hopefully they will agree, to have training and institutional review, networking visit in September and October in West African countries, starting from Mauritania all the way to Cameroon and Gambia and Senegal and Ghana and others. So that we are planning to do. And this is, of course, a very important event also. Patrick Steele was here last year, and he gave a related workshop on drug discovery and development. And today, we're going to hear a lot of parasitology and lots of molecular biology and lots of X-ray, lots of structural biology of the disease. Now, I just would like to conclude my comments by saying, when you say neglected tropical diseases, by all definition of the word, they are neglected. neglected that they are among the poorest people of the world, where the prevalence is extremely high in topics of America, Africa, and Asia. The most impoverished communities, neglected in terms of investment in healthcare, prevention, and treatment, neglected in terms of science. So very little is known about the drug targets and others. And in fact, this network which was created under the leadership of Durham University was precisely on identifying targets. So the least investment in terms of science of diseases are neglected topics. And can you believe it? It really affects millions of people, if not billions. So they are affecting a lot of people, and their disease burden together is much larger than cancer and other diseases. So they are neglected by all definition. Pharmaceutical companies are not very much interested. If they will do that, they will do it as part of their corporate social responsibility, because they know that this is a poor man disease and they don't want to invest in any disease which would not give them. Having said that, we have this opportunity of learning from great people, not in terms of their knowledge and excellence, but also in terms of their mission and their passion for that particular. And they are very much into it. They are global citizens. They are everywhere and trying to help people who would like to focus their research. So apart from the understanding of the diseases, and apart from networking which you would have with them, I think the outcome would also be if you can focus your research, at least part of it, towards these diseases, to have a better understanding, more attention, and eventually to have a cohort of people who are interested in these diseases, because they are our diseases. Poor people of the world. Thank you. Thank you very much.
Thank you very much, sir, for your insightful remarks. Next, I would like to request Professor Dr. Paul Denny to his talk. We will be talking about leishmaniasis, drugs and diagnostics for our disease. What you say?
Use episodes on slides and the next policy. Yes. Thank you. So,
We have 76 people online.
Yes, sir.
Well, it is immensely popular lecture pod. You were a celebrity, now you are.
well, yeah, well, thank you, Iqbal. Thank you, my friend, for that very warm welcome. And I should say that this country and its people are now very close to my heart, and a lot of my work is. So, Iqbal has very eloquently introduced the disease of interest, which in my case is leishmaniasis, and particularly cutaneous leishmaniasis. Now, my background in the last 15 years really has been in drug discovery, a very, very difficult process, which I'll take you through. More recently, working particularly with some of there is to think about diagnostics, and we've now got projects which involve Pakistan and the ICCBS in diagnostic development, particularly to that exist in Pakistan as well as in other countries. So I'm in the Department of Biosciences at Durham University, and with Emka over there, I'm part of the biomolecular interactions group. So we have a molecular approach to science. It's not working.
Should work, right?
There we go. So just an introduction, because I doubt many of you have heard of Durham, let alone been there. So I'll give an introduction. So it's a medieval city. So the cathedral there is approaching 1,000 years old, which is very new in Pakistan terms, obviously. You know, you are one of the cradles of ancient civilization. The castle there was built at the same time by the Norman invaders who invaded from France. Symbols of power as well as religion. Now, the university sits partly within the castle, but also within rather more modern buildings as well. And it's rated 78 on the QS global rankings at the moment. It has about 20,000 students, 27 departments, and remains a top institution in the UK and globally. So a good place to be. But, you know, we do look globally, or at least many of us look globally. There's a local and an international aspect to our work. So, you know, we're right there in the northeast of England, just south of Newcastle upon Tyne. You may have heard of the football club, Newcastle United. We're not that far away. So, but as Iqbal was saying, we led a network which was purely based on science and equity. So it was a completely flat line. We weren't leading it from the top. We were leading it on a horizontal structure. So Pakistan was a very important partner, Uruguay, Argentina, Brazil, and India. So pretty much covering a very large part of the world. Now, that was focused on leishmaniasis and Chagas disease, which is caused by Trypanosoma cruzi and is very prevalent in large parts of South America, not elsewhere. But I'm going to just talk about sphingolipids here. For those of you that don't know, sphingolipids are very, very important lipid species, part of the structure of our plasma membranes, involved in everything from trafficking of proteins and other lipids, signal transduction, and the formation of these lipid rafts. Who's heard of lipid rafts? Anybody? No? All right, okay. Well, lipid rafts are little domains that have been identified in our plasma membrane, which encircles our cells, and sphingolipids are an important structural and formative feature of these domains. And they're involved in numerous things, but probably most particularly signal transduction, how we communicate signals from the external environment of cells into the internal processing. So very important. Now, before I talk about leishmaniasis, I'm going to talk about toxoplasma. So toxoplasma is another protozoan parasite. It's a parasite of any nucleated cell. Now, there's a lot of lamb eaten in Pakistan, I'm aware, and toxoplasma is a parasite, not just of people, but of lamb and many other animals as well. It can infect any nucleated cell. So consumption of lamb is likely to lead to contact with toxoplasma. Now, this doesn't mean that everybody is sick, but it probably means that about half of the people in this room have encysted toxoplasma within their bodies. Not normally a problem. The definitive host is the cats. That's where the sexual cycle takes place. But it's not necessarily essential for the transmission of the parasite. It can be just a pure food to human transmission. It causes encephalitis and other neurological problems if ingested when a woman is pregnant, particularly in the third trimester. And it's related to the Plasmodium species that causes malaria, much more famous parasite, of course. So we tend to use it as a model species to study biochemistry and enzymes. So just to introduce you to sphingolipid biosynthesis, an enzyme, serine palmityl transferase, or SPT, is the first and rate limiting, the controlling step in sphingolipid biosynthesis. And I realize there'll be a number of people familiar with chemistry, probably more familiar than me. So we have palmityl coenzyme A here, and we have the amino acid serine here, and the serine palmityl transferase mitigates the conjugation of these into 3-keto dihydroxysphingosine, or KDS, which you can see there. That's the first step in sphingolipid biosynthesis. Okay, so characterized by this amide linkage, which you can see here. So that's the base of sphingolipids. Now, in us, in humans, and in almost every other eukaryote, we thought every eukaryote until a few years ago when Emka and I actually worked on the apicomplexan, the toxoplasma and related parasite enzymes. Every other eukaryote has a dimeric core to this enzyme. So there's 2 proteins that are involved in the structure and function, as well as some smaller accessory proteins, such as this one here, TSC3. So the primary ones are LCB1 and LCB2. The names come from sort of yeast genetics, essentially, like a lot of eukaryotic names. So most of them, they essentially have LCB1 and LCB2. They're transmembrane enzymes, and they have to be together for functionality. LCB2 has the catalytic function. LCB1, a different protein, related but different, associates with it and is thought to regulate function. It's a rate limiting step. So recently, very recently actually, this was actually elucidated. The structure of this was elucidated by cryo-EM, and I think Emka will mention cryo-EM to some degree. Yeah. So in toxoplasma, we chose to study toxoplasma because it's relatively straightforward to manipulate. We identified the enzyme on the basis of homology. I won't go through that. We localized it to the endoplasmic reticulum. And you can see that's a marker. That's the toxoplasma serine palmityl transferase, as we called it, located there. And then biochemically, by expressing and isolating the protein, we showed that it was functional in vitro. And there you can see by mass spectrometry, this peak is KDS, the product of serine palmityl transferase action. Now, I mentioned the eukaryotic serine palmityl transferase has 2 subunits. It's heterodimeric. In contrast, the bacterial version is very different. It has the same function, but it's homodimeric. Okay, 2 subunits, but they're exactly the same. It has to be dimerized to function. And with Emka, this was published back in 2017. We identified that the apicomplexan serine palmityl transferase is basically like this bacterial version. It's related, it's evolutionary derived from that particular structure. So it has a very unusual origin. So thinking ahead in the pathway, that we spoke about serine palmityl transferase and KDS, but the base unit of all complex sphingolipids is ceramide. So there you can see the amide linkage here, and there's the ceramide. There's 2 fatty acid or hydrocarbon chains, and then just point out this primary hydroxyl group here. So in our cells, we have sphingomyelin. That's about 10% of our plasma membranes. And then with sphingomyelin, you can see phosphocholine. There's phosphocholine added to this primary hydroxyl group. But in plants, fungi, and indeed, as I'll show you, leishmania, and to a degree, toxoplasma, We have phosphoinositol. You see this inositol group here, very different from the choline. So we have very different phosphosphingolipids in different species, in different kingdoms of life. And of course, we have different enzymes. So I'll come back to that. And other work that both Emka and I have been involved with, this is very recently published. It was actually in 2023. Just took a long time to come out. Is the ceramide synthase. So this is the enzyme responsible for the production of the ceramide species, that base unit sphingolipids I mentioned just earlier. Now, again, in a quirk of evolution that is very specific to the apicomplexan parasites, including Toxoplasma and Plasmodium and many others, there are two isoforms, ceramide synthase 1 and 2. We name them imaginatively. But there's a whole group in ceramide synthase 2 and its orthologs in other related parasites, which are non-functional. And we spend a lot of time showing this biochemically. And we also manipulated the parasite to examine the effect of deletion of this enzyme activity and this active one as well. And what we can see is that deletion of ceramide synthase 2 has a bigger effect on fitness, the ability of the parasite to disrupt cells. As you can see here, this is disruption, see plaques, and this is non-disruption of a parasite that has lost ceramide synthase 2. So it's very important for fitness, but we don't quite understand why. Okay, so that's just really an example of some of our work to give you an understanding of sphingolipid biosynthesis and its importance to the parasite. But we're now going to move on to leishmaniasis. So all of you will be familiar with leishmaniasis because, well, I hope you are. Are you?
Yes.
Yeah, all right. Because, you know, it's a major disease. As Iqbal said, Pakistan here has one of the greatest burdens of cutaneous leishmaniasis in the world, but it is a disease that is spreading very rapidly across all continents, including Europe and North America. So let's think about the life cycle. If you're not a person that works on parasites or leishmaniasis, you may not consider life cycles important, but this is the sort of basis of understanding disease. So sandflies, which are about 2 or 3 millimeters long, tiny, tiny insects, transmit these flagellated promastigote forms into humans or other mammals. There are other mammals for most of the species of leishmaniasis. They invade macrophages, part of our immune system, and we get disease here. So this is a boy in Sudan with so-called visceral disease. So that's infecting his liver and spleen. This is a man in, I think that's Colombia, and you can see the destruction of his nasal tissues. So that's caused by Leishmania brasiliensis. That's mucocutaneous disease. And this is what you'll see in Pakistan. This is a relatively mild lesion. You'll see a lot worse than that in the outbreaks which are ongoing. And this is cutaneous disease. Okay, and in Pakistan, that's caused by Leishmania major and increasingly problematically with Leishmania tropica. And then the cycle begins again with the parasite being taken up by a sandfly bite and then going on through its life cycle. Now, current treatments, they're not good. This is how I and many others got involved in drug discovery and sought to engage industry in that process, which, as Iqbal mentioned, there's been a reluctance for industry to spend money on this disease of the very poor, but there are some initiatives. So in Pakistan and many other countries in the WHO Eastern Mediterranean region, which have major problems with cutaneous disease, you'll have these antimonials used. And I'll show a picture of the administration of these in a little while. So they have antimony in the middle, very toxic, unpleasant. They're injected either directly into the lesion or systemically very toxic, lots of side effects. That's the first line therapy. We have paramomycin. That is used as a second line therapy at the moment, along with miltefosine for visceral disease, largely. Liposomal amphotericin B. This is primarily used in the elimination exercise, which is in India, Bangladesh, and Nepal, aimed at eliminating visceral leishmaniasis caused by Leishmania donovani, and it is having some success. We have pentamidine as well, but pentamidine is so toxic it's generally being taken out of use. So we have a very limited arsenal of drugs, and it's only miltefosine that is available in tablet form, only that's already available. Okay, so we're interested in how these drugs work, of course, because it's very unclear. So I've been involved in studies and remain involved in studies looking at how miltefosine, this orally available anti-leishmanial, actually functions. But I'm going to just talk about a sort of baseline drug discovery process we undertook with GSK in Tres Cantos, Madrid, targeting the enzyme IPC synthase that catalyzes the production of inositol phosphoryl ceramide, or IPC. Remember, that's the complex lipid species, the phospholipid species that is different from sphingomyelin that we have in our cells. So we have sphingomyelin synthase, which takes phosphocholine from phosphatidylcholine, the lipid, and places it on ceramide, whereas Leishmania, as well as yeast and plants, have IPC synthase to take the phosphoinositol. Okay, so different enzymes, different products. That indicates an important niche that we can target with selected drugs. So the theory behind being able to target this and have a rapid effect and parasite killing is based on this. So the action of the enzyme, we have phosphatidylinositol, a common phospholipid. The phosphoinositol is taken and is added onto the activated histamine of the enzyme, which you can see here. This releases diacylglycerol, DAG. That's a growth promoter, that's mitogenic. That's well known in mammalian systems. On the other side of the coin, this is a very labile structure. It doesn't exist for very long. And we have ceramide coming in. Remember, I pointed out that primary hydroxyl group. It comes in and it steals the phosphoinositol from that enzyme, producing the mature product, IPC, the important mature sphingolipid that is in the membrane of the parasite, and releasing the enzyme. So that diminishes the amount of ceramide, which is cytotoxic. Ceramide is cytotoxic, it's known to be pro-apoptotic in mammalian cells, and indeed in plants as well. Okay, so this enzyme sits at the tipping point of life and death. So if we interfere with this enzyme function, we can promote death. So that was our theory in approaching this. So this has already been validated in fungal systems. So yeast and fungi have the IPC synthase as well, known as AUR1, AUR1. Number of natural products, most predominantly this depsipeptide, circular peptide, aureobacidin A, which you can see here. Okay, but the enzyme is a very complicated enzyme. It has 6 transmembrane domains. It has membranous substrates and a membranous product as well. So it's complicated to assay. So if we want to actually do a screening exercise, we have to think about how we're going to deal with this very complicated transmembrane enzyme. Can you develop a high throughput screening assay for this very complicated transmembrane enzyme? Now, we do, and you can look at such enzymes in vitro, in a tube, in a lab, by expressing them into proteoliposomes. Okay, these are lipids and proteins. They're expressed together, so we have a functional transmembrane protein, and then we can assay them. But it's expensive and it's complicated. So really, in terms of screening compounds, we need to think very clearly about how we can develop an assay system to screen not just tens of compounds, but hundreds of thousands, because an industrial drug screening program is hundreds of thousands of compounds. And I'm also going to show how we screened about 2 million compounds against this enzyme. Now, I already mentioned that yeast OR1 is the yeast IPC synthase. So what we did is we had a yeast cell line, and this is your baker's yeast, Saccharomyces cerevisiae, very easy to work with, cheap, safe, quick growing. And we support growth of this essential enzyme by having the OR1 enzyme on a selectable plasmid. So URA3, that's a marker that allows for growth in the absence of uracil, which is essential in this line in the absence of that marker. And then we take another plasmid, which has the Leishmania IPC synthase encoded in it under the control of a galactose promoter. So that promoter is only activated and protein expression only happens when glucose is down and galactose is up. And that's selected by this marker LU2, allowing the yeast to grow in the absence of leucine and the amino acid. So we have this heterozygous system. So we have both plasmids, both enzymes there. So if we reduce the glucose and add galactose, we get expression from here, and we get expression of the Leishmania enzyme. And then if we add this agent here, 5-fluororontic acid. Now that's a substance that produces a toxic byproduct in the presence of URA3. Okay, so you have a negative selection there in the presence of 5-fluororontic acid. Remember, we've got galactose, so the Leishmania enzyme is being expressed to support growth. So a negative selection, so we lose the OR1 on this URA3 plasmid, and we end up with a yeast strain that's completely dependent on the expression of the Leishmania IPC synthase. Okay, this is when we go into the not just high throughput screening, this is ultra high throughput screening. So you may be familiar with 96 well titer plates, but these are 1356 well, 1356 well. So these are nanoliter plates. So all of these plates have nanoliter quantities of the yeast growing. So there's a lot of work that went up into the assay preparation and validation. And then it may be a surprise to you that the actual process of screening 2 million compounds is quite quick. It's really the assay development that takes the time. So after a primary screen at a single concentration, then there was a secondary screen at 2 concentrations. And we screened against our Leishmania yeast, but we also screened against yeast supported by its own enzyme, OR1. We wanted to know which of the compounds was specifically inhibiting the actual Leishmania enzyme, rather than just being generically antifungal, anti-yeast. So then we end up with 4,000 hits. So these are active and specific compounds. And at that point, we can do dose response. We can generate IC50 curves. So we can work out which is the concentration of compounds that inhibits 50% of yeast growth. Now, if we look at this here, these are all log values. So 6 is 1 micromolar, 7 is 100 nanomolar, and 5 is 10 micromolar. So we have the IC50 values for the yeast OR1 here, and the IC50 values for the Leishmania IPC synthase here. So if we add the color to it, the more selective ones are towards this corner. So the more exquisitely active against the Leishmania and the less active against the yeast enzyme, all in this corner. And again, this is a log value. So all of these, that's a log, negative log 10, 1.5. That's a selectivity of about 33 or 4. So taking that as a cutoff, we have 500 compounds. So actually, in a matter of a few weeks, we've gone from 2 million to 500 compounds, which is then a kind of manageable value for more complicated assays, such as the assay we developed to study the enzyme. I already mentioned it's a complicated transmembrane enzyme. So we developed this by taking membranes from yeast and refining them, washing them, formatting them, putting them in a 96 well plate assay. I can talk about that later as you wish. And we add a fluorescent MBD ceramide, fluorescent shown here. We add the enzyme, we add the phosphatidylinositol, which is the other substrate along with ceramide, and then we get a fluorescent product, fluorescent IPC, which we can measure in a plate reader. Quite simple, although the assay is very complicated and it caused us a lot of problems. But some of the compounds we identified, such as the benzazepanes, which we took forward, demonstrated themselves to have the low 1 micromolar activity, so nanomolar activity. So this connected our yeast assay with genuine enzyme inhibition. And we published this in 2018. So that was good. And also tamoxifen, which you may have heard of as a therapy for breast cancer. Our colleague Sylvia Uliana, who is an old friend and colleague now at USP, which is in Sao Paulo in Brazil, has worked on tamoxifen as an anti-leukemia for many years, has actually initiated clinical trials as well. And we showed that tamoxifen does indeed inhibit the inositol phosphoryl ceramide in leishmania. Now, on the final part of this, another repurposed compound, clemastine, which is a well-known antihistamine. So this is the result of a very long project, actually, with Patrick Steele, who, as Iqbal mentioned, was here last year. And we showed that it inhibits the enzyme, but we took it a little bit more further then. We took it into animals. So we can see clemastine is in green. So it reduces the lesion size, which you can just about see here on the animal. Reduces the parasite load as well, which you can see here. And importantly, if we take a broad mass spectrometry metabolomic approach, it's hidden behind the little banner here, but inhibition of that enzyme also causes a very dramatic increase in ceramide. So this gets back to the balance between life and death. Remember, I said that we targeted this enzyme because of the balance between diacylglycerol and ceramide, or life and death. So this promotes an increase in ceramide, which we connect to the death of the parasite in our other assays. And it made the front cover, which was nice. Okay, so we have to be very, very critical of ourselves in our work. This, science is a process of iterative criticism of your own work, iterative understanding. So is finger lipid biosynthesis actually a drug target? Now, obviously I'd spent years pursuing it as such, but is it? We have to reevaluate. Now, in earlier work, LCB2, that's the SPT subunit, which I mentioned, which is not like the toxoplasma, it's like human, can be knocked out in Leishmania major. So this is some work I did myself, and this is from another group. Now, when we do knock this enzyme out in Leishmania major, remember that's one of the core causes of cutaneous leishmaniasis in Pakistan. What we don't see a loss completely of pathogenicity, we just see a delay, consistent delay in pathogenicity. So what does this mean? I mean, does it mean that sphingolipids are not important for disease and pathogenicity? Does it mean they're not a drug target in reality? Well, then we looked closely, and you're probably all aware of whole genome sequencing becoming very common using Illumina technology, and the way we can relatively cheaply now generate a whole genome sequence and then analyze it. So what we chose to do is actually take this line here and the controls and the whole genome sequence. And what we noticed, yeah, sure, they'd lost the LCB2 subunit, the SBT catalytic subunit, that was clearly gone. But they'd also lost several other things and completely lost a gene here, which you can see the gap between the parental control and the knockout, completely gone. We hadn't targeted this, it just disappeared. And this is a gene encoding a putative ABC transporter, which is most closely related to an A3 type, which is involved in humans in sterol, cholesterol trafficking. So then we tested the effect of the loss of this in a more friendly system. So then we turn our hands to Leishmania mexicana. So that's not really a very important pathogen, but it's rather easy to manipulate. And then we have the CRISPR-Cas9. Have you all heard of CRISPR-Cas9? Yeah. So CRISPR-Cas9. So we can relatively now quickly knock things out. All the work I told you was using homologous recombination, the previous studies. It took months, essentially, but at least 3 months before you had a knockout of the gene target. So now you can achieve that in 2 weeks. It's quick and effective. So using CRISPR-Cas9 in Leishmania mexicana, we could not knock out the LCB2 subunit. It failed, unlike in Leishmania major. However, if we knocked this out, this putative ABC transporter at the same time, we could remove it. So it appeared from that we had a system of compensatory loss of this gene, allowed loss of the LCB2 transporter, the LCB2 SBT subunit. Okay, so it's a complicated system. So this data indicate, yeah, well, maybe LCB2 is essential. It's just that we're looking at parasites that have lost other things. So we published that in 22. Now, what about IPC synthase? After all the effort, what was it worth it? You should always ask, is it worth it? They're doing exactly the same kind of thing, taking the CRISPR-Cas9 approach in Leishmania mexicana. We can knock out the enzyme really easily, which I thought that's a bit perturbing, a bit problematic. This is in insect form promastigotes. But we do lose the IPC. This is again mass spectrometry data, specifically looking at the wild type, the knockout, which shows nothing, and then when we add back the gene for IPC synthase. So we see we have lost, for all the species of IPC, we see we have lost that in the knockout species and recovered it in the add back. And then we go to an in vivo model. So we look at mice in this case, valve C mice. And yeah, we do have a fitness cost, okay, which is good. So these are the parasite burden per unit weight of tissue removed from lesions, tail lesions in this case. So we can see we have a 10-fold decrease in the quantity of parasites recovered in the lesion material for the knockout. And we have no significant lesion at all, actually. The parasite hasn't gone away, but it hasn't caused pathology. Curiously, and we can talk about this later, the add back did not recover the parental phenotype. It remained no lesion, and low parasite quantity. So it's a complicated picture. Similarly, and this is work we did with Santuza Teixeira in Belo Horizonte in Brazil, working with Trypanosoma cruzi. It's a parallel project, very recently published. Ours is still in preparation. Exactly the same phenotype as this as well. So the enzyme clearly has important functions in both Leishmania and Trypanosoma cruzi. So just to, for Sama's benefit, we also work with natural products occasionally. So we've looked at natural products derived from a fungal extract library, saponins from plants, semi-synthetic. We're still working on those. And chalcones, again, this is Patrick and another colleague from Rio in Brazil, Batira Rossi Bergman, working with chalcones, again derived from plants. And we have looked at aureobacidin and found it has quite acute activity against toxoplasma, actually. But it does not target sphingolipid biosynthesis. We spend a lot of time showing that. So what next? I mean, it's a complicated process, drug discovery, but we have to pick up learning as we go. We cannot just keep banging our head against the door, doing the same thing. So again, we work with GSK and the Transcontas Open Lab Foundation. And also, at the time, we were able to gather co-funding from our GCRF network that Iqbal mentioned that brought us all together in 2018. So we worked with GSK, Transcantos, and they had a number of compounds which had been identified from a mass screen they'd done against Leishmania donovani, Trypanosoma cruzi, and Trypanosoma brucei. And they're particularly interested now in those that target the cruzi, anti-Chagas components, as well as anti-Leishmanial compounds. Okay, so then we had this co-funded project with them, which is still ongoing. So it involves several scientists in the UK, Ashish in India, Iqbal here, Santuza, who I already mentioned in Brazil, and Carlos Ravello, who's with me in Uruguay at the Pasteur Institute in Montevideo. So you don't have to read this, you'll be glad to know. But we were given 17 compounds which were identified as active, particularly active and selective against the Leishmania donovani in the study I mentioned, and another 8, which were active against Trypanosoma cruzi in the same study. And these were all passed on phys-chem properties as developable. So they're not the ones that would be kicked out. So you'd be kicked out if you had a nitro group because of toxicity, you'd kick that out. So these are all relatively small, not particularly polar, not particularly lipophilicity, and rather water soluble. Okay, so these pass all sorts of medicinal chemistry criteria through the medicinal chemists actually at GSK. So then we went through a process of triage. Now, again, it's very important to really strictly stick with triage. And in academia, we tend to not really do triage. We tend to just, as I said, bang on and push ahead, even if some of the indicators are wrong. But when you work with industry, you have to be very, very strict about the triage process. So as I mentioned, we started with 17 compounds selected from the Leash and Chagas boxes. And remember, these were ones that were identified in assays against Leishmania donovani and Trypanosoma cruzi. So we looked for activity against Leishmania mexicana and major. Okay, they're our target species, and T. cruzi. And we ended up, because of activity across all of these, we selected 5 from leash box and 1 from Chagas box. But quite quickly went from 17 to 6. And then we generated resistant lines against the Leishmania species and the Trypanosoma cruzi species. This is by in vitro evolution, increasing the compound quantity to generate resistance in vitro in multi-well plates. So 5 compounds generated significant resistance. And then we're down to 4 compounds which show no evidence of sharing a mode of action with any of the clinical drugs. Okay, so we cross-referenced with the antimonials, miltefosine, etc. So we've gone from 17 to 4, all with the coronavirus pandemic in the middle, actually. But anyway, we carried on. Phase 2, and this is what we're in at the moment, is the thermal proteomic profiling. So we can talk about that in detail out with this, but essentially that's looking for protein compound engagement throughout the whole proteome. So it's a complicated process, it's quite challenging, but we're undergoing this at the moment. And we've completed metabolomics and lipidomics. It's a bit like the study I showed you for cloustine. We're looking for the effect of these compounds on small metabolites and also macromolecules such as lipids. Okay. So by bringing together the genomic data, which we'll get from these resistant lines, the proteomic data and the metabolomic and lipid data, we are starting to see patterns and things that we will report very soon. Now, actually, Emka and I sort of devised this as we were putting together this particular application about how we would triage, again, triage is important, proteins. Okay, so if we didn't know the function, we would deprioritize. Now, you might ask why. Why would you deprioritize? Well, if you don't know the function, it's very hard to create an in vitro assay so you can actually test the compound against the protein in vitro. And maybe, you know, putative but unconfirmed function, yeah, we may take that forward. But likely, yeah, probably would take that forward, but confirmed function we would prioritize, because then hopefully we can actually get an assay and we can get some understanding of the way the compound interacts with the protein, which is something Emka is going to talk about in some detail after lunch. Further prioritization, this is when it comes around to really protein structural concerns. So we really do need to get a high order level of understanding of the way the protein interacts with that ligand, that compound identified. So something that's disordered, that's impossible. It's just a length of string and a molecule. You're never going to understand it. Integral membrane proteins, you might be surprised that I would put integral membrane proteins as a low priority, having, you know, worked on IPC synthase and others, but they're very complicated. It's very difficult to get, even now, a high level understanding of how a compound would interact with an integral membrane protein. Soluble proteins, yeah, we want soluble proteins. We want particularly soluble cytosolic proteins. Okay, they're most likely to be druggable behind less membranes. If it's a known or obvious drug target, yeah, that's not a bad thing at all. That's good. But even more important is a new target, something that has not previously been studied. So that's our list of prioritizations. So, but even with this prioritization pipeline, although we are seeing some progress in the project that we're running at the moment with Trescantos, it's very high risk, very, very high risk, and expensive of time and people and money. So we really need to think also about other solutions. Now, this is where my interactions and trips to Pakistan become very important, actually. And I've learned really that drugs are not the only answer to the problem. There are other things we need to consider when thinking about, in Pakistan, cutaneous leishmaniasis. This is where I want to come on to diagnostics. So one of the things about not just cutaneous leishmaniasis, but leishmaniasis in general is it's caused by lots of different species. The most complicated is cutaneous leishmaniasis. There's multiple species. Overall, there's more than 20 species that cause leishmaniasis of all forms. It's complicated. And not all species respond to therapy in the same way, so not all the drugs are effective. And also there's misdiagnosis commonly in cutaneous disease, everything from leprosy to skin cancer has been misdiagnosed. And that's a problem. So diagnostics are a real key part of the control armory. We need to know what we're dealing with. Now, at the moment, we have visual clinical inspection. There are some very, very experienced clinicians out there, including in Pakistan, who can look at a lesion and know whether it's leishmaniasis or not. That's not necessarily the case where the patients are in very rural areas. A definitive diagnosis at the moment is a biopsy. So this is a lesion biopsy, and these are amastogotes, the pathogenic form within a phagocytosome in a cell taken from a lesion biopsy. But that's obviously quite technical. You need a microscope, you need training, you need a lesion biopsy. And also this can be backed up by PCR, which is again complicated. As far as rapid diagnostic tests, we all became very familiar with RDTs during the COVID-19 pandemic. Those little cassettes that you would get, various types around the world. They do exist for leishmaniasis. There's one for visceral leishmaniasis that's been introduced. This is for Leishmania donovani leishmaniasis, but it detects antibodies. Now remember that the COVID-19 diagnostic, the lateral flow test, detects protein. So there's some concerns about, the state of infection when you detect that protein, but it detects protein, so it indicates an active infection. Detecting antibodies does not necessarily indicate an active infection. Could be historic, limited value. And more recently, we've had and actually this is a commercial, this is a non-profit making. We have CL Detect from a US company. Now, this is FDA cleared, but in my view, is rather limited in terms of the clinical trials that have been initiated. So this is based on a protein, so it does detect active infection, but it cannot distinguish between causative species. So it cannot tell you, in Pakistan, for example, whether it's Leishmania major leishmaniasis or Leishmania tropica, or indeed a co-infection with both. It does not give you that. So it needs to be backed up really by a PCR-based diagnostic, which of course is laboratory intensive, technical and expensive. You need staff, you need a PCR machine, etc. So Iqbal mentioned the opportunity, and I'm very grateful for this opportunity I had on a trip in 2019 to visit the Jinnah Hospital in Karachi and actually meet a family with their 4 children. Their father brought them in. They'd been to visit, I think, his parents in northern Pakistan, and then come back, and the children, who were anywhere between, I think, 3 and about 7 or 8, had all developed classic leishmaniasis lesions. And a great team at this hospital led by Dr. Cozy. And so they were treating these patients, and the father, the children and the medical team gave us a chance to speak to them.
So you can see the lesions here. This one here is healing. This is a young girl, but you can see the scarring there is going to be significant. Healing here, again on the nose, that's going to be significant scarring. Less well healing and really not healing at all on this boy's nose here. Now, the healing or non-healing, these are all these children receiving very painful treatment with antimonials. This is probably, in this case, related to the species of parasite, because Leishmanium tropica tends to lead to chronic, non-healing infections that don't respond well to treatment with antimonials. And even in the UK, so this person here got cutaneous leishmaniasis in, I think it was, a very fancy holiday backpacking in the jungles of Central America. So on the upside, she has now become a leishmaniasis advocate, which really does give us a little bit more traction in the West, in Europe and the US, for people to understand and recognize the problem that exists in places like Pakistan. Now, I already mentioned the problem of antimonial treatment. So this is an injection directly into the lesion. These are large volumes, up to about 5 mils of toxic antimonial directed into the lesion. And this generally works with Leishmania major infections, but we're getting close to 50% of treatment failure with Leishmania tropica infections, including in Pakistan. This is a real problem. This is why in the hospital case, there was one of those children not responding to therapy, and the lesion was becoming chronic and problematic. So, just to finish up, I'm not sure how much time I've got. How much? 2 minutes. Okay. So we are thinking about diagnostics, and we are basing it, and again, we have to look at the instruction we get from the community and industry where appropriate of how to do things. So we look at the Reed-Leash group, which is the Drugs for Neglected Diseases group, which is looking at diagnostics, particularly for cutaneous leishmaniasis, where the problem is acute. So they have agreed with us, actually. We had started this work before this manifesto was released. That LAMP, loop-mediated isothermal amplification, was a process that we could exploit for an appropriate diagnostic, particularly one that would distinguish between different species like Leishmania major and Leishmania tropica. The advantage of LAMP, it has a complexity to it, which I won't go into, is it's isothermal. It's a single temperature. So typically around 60 degrees, typically around 10 or 20 minutes, you get a signal. So we are working on developing this as a tool. So we have already established sets of primers that are distinguishing between different species, tropica and major. It's all based on a fluorescent signal. Now, importantly, Zesis Koussogiannis, who came with us to Karachi, is still in Karachi, and he is actually road testing our primers against some clinical isolates from Sindh province, which is very important that we do. So in this, we're working very closely, as I mentioned, with SAMA and the ICCBS in Karachi, and also very closely with Marlowe's Peters group in Newcastle, soon to be in Manchester, because not only do we want a set of primers and an assay that works and is robust, but we also want a tractable instrument, a box to put our tubes in, so we can actually do the assay in a field station in Pakistan, for example, in a rural area. Not a doctor's surgery, but a paramedical unit that has to do the initial validation and diagnosis of the patients. So we're thinking on reality. So Iqbal mentioned, there he is, mentioned this meeting that we held in Nairobi in February. And really, we are stronger together if we do this. We are nothing without our colleagues in countries like Pakistan. And just thanks to everyone. And if you like, you can follow me on Twitter and find out what's going on a day-to-day basis. So thank you very much.
Thank you very much, Dr. Paul. So we have the next 10 minutes for question answers. So if anybody has any questions, you may ask now. Any questions or any comments?
Thank you very much, Dr. Paul, for our talk. I just want to know, is there any vaccines available against this parasite?
It's a good question. There's currently no vaccines. Now, there is an effort, an increased effort, I'm told. There was a very small number of people working on, shall we say, defined vaccines based on proteins. There have been clinical trials, but there's no success and no vaccines on the market in the clinic up to this point.
Any other questions or comments?
Thank you, sir, for the presentation, sir. Is there any phage therapeutic used in treatment?
Well, the phage are completely directed against bacteria, so they would not work on a protozoa and eukaryote, such as leishmania.
Sir, firstly, it was a nice session, and I would like to know, at least people have some vaccination and therapeutic facility for that. How can we cope up with this? Any preventive measure should be take or something like that?
Sorry, say that again. So I got the thing about vaccination. What was the other bit?
Despite there is no vaccination or therapy we can use for that. So any preventive measure should be taken to prevent the leishmaniasis or something.
Well, yeah, I mean, that's interesting. I would say that it's a tragic disease because really one of the main drivers of whether you become infected or not is your nutritional state and your health as a background. Probably if I was to go to Sindh province and get bitten, I'm fairly unlikely, to be honest, to get cutaneous or any other leishmaniasis. So, but if you are malnourished, you have a much greater chance because malnourishment, as you probably know, leads to immune suppression. So I think we don't really understand the breeding sites of sandflies very well. Bed nets, yeah, kind of work, but they're so small. If there's any damage to the bed net, they can easily get through. And also, people are bitten in the fields rather than anywhere else. It's a very rural disease. But it's a good point. But I would say that ensuring good health and general health care, which is challenging. Yeah.
Curious?
For your question or comment, please.
I'm Dr. Fawaz Rashid from Shifadari Family Millet. I just want to know, do we have cell line data, human cell line data related to ceramide?
Cell line data, what do you mean?
Any cell line study on ceramide related to human cell line?
Oh, do you mean the enzyme or the molecules?
Let me clarify the
Yeah.
questions. Actually, the ceramide is supposed to be cytotoxic.
Yes.
What does it have the implications in the particular online investigations that whether it is cytotoxic, it is supposed to be toxic for the human as well, human cells or not?
Yeah, I mean these studies are very unclear in leishmania, but in human cells, if we induce ceramide to be overpresent, to increase, then it does for sure induce apoptosis. It's known to be a secondary signaling molecule in the apoptotic process. That does not mean that in a parasitic infection, that if we increase the ceramide in the parasite, this would lead to a problem in the host. No, it's much broader than that. We can be quite specific.
Does it mean that there must be a balance between the ceramide and the, you mentioned phosphoglucolipids, I think, if I, not, phosphosphenol.
Oh, the diacetyl
There must
glycerol.
be a balance between these.
Yes, that's well understood. It is a balance, and the sphingomyelin synthase in human cells is one of the enzymes which has a key function in the balance between those two molecules. It's not the only one. It's a complex process which from the top of my head, I'm not qualified to talk about.
So I want to have a bit clarity more.
Yeah.
If you allow.
No,
So
please.
if you are going to target the ceramide. So are we considering this balancing, or so the care must be exercised between the regarding the balances of this, because we are targeting, if we are targeting the inhibitions of this. So how can we able to make this balance of this process of apoptosis you mentioned, balancing of this too?
Yeah, well, I think you're asking whether there could be toxic effects
Yes.
of targeting the parasite, and increasing ceramide in the parasite may have a toxic effect on the host. This would only, if you had a very specific drug that only targeted very specifically the IPC synthase in the parasite and led to a very specific increase in ceramide in the parasite, then you would not target any other human cell. You would only target parasites within infected cells. And given the parasites are approximately 1/10 the size of the cell, and the amount of ceramide in them is quite small, I don't think you'd have a great effect on that cell. But even if you did, apoptosis of an infected cell is a positive thing. That's one of our immune responses. And Leishmania have a very sophisticated process of preventing apoptosis. So there's no danger of broad cytotoxicity, as long as the normal processes of safety is considered. Yeah.
Thank you.
So we have a question from one of our online participants. If you allow.
Of course, yeah.
Okay, so somebody's asking how many research groups from Latin America, especially Brazil, are involved in this project, as the disease has long been diagnosed and worked through so far, and what are the progress made so far?
So with our GCRF project, we had, I'm trying to think, 1, 2, 3, 4, probably 10 groups across Latin America involved in the project at the start. And there is a great focus in Latin America, particularly on Chagas disease. And we continue to work with colleagues there, particularly Carlos Rebello and Santuza Teixeira, on the Chagas disease element of this. But there's also leishmaniasis as well. Brazil has a burden of both visceral and cutaneous disease, which is extensive. So they remain important, but also the parasite species causing leishmaniasis in Brazil are very different from those causing leishmaniasis in Pakistan. And the approaches for therapy are going to have to be different. The diseases take a different course. They respond differently to therapy. So we need to consider all those when we actually consider drug discovery, or indeed even diagnostic development as well. I'm not sure that answered the question, but I did my best.
Thank you, sir. And we can take one last question from another online participant. And I think you've already covered this in your presentation. To what extent success has been achieved in understanding and controlling leishmania, and what future prospects do we see in terms of curing it through a natural products? Any insights?
Okay, so with visceral disease, at least caused by Leishmania donovani in India, Bangladesh, and Nepal, we will probably see great progress. We are seeing great progress through that liposomal antimonial therapy drive. For visceral disease caused by Leishmania donovani in East Africa, no progress at the moment. And the political situation in a lot of East Africa is making that very difficult as well. Cutaneous disease, rising. There is little progress at the moment. It is becoming a problem. It's rising in Pakistan, but it's also rising across the globe. So I don't see any progress. As well as natural products, well, I'd encourage people to continue to seek therapies from wherever they can find them. There are two natural products, ampicillin B, which is very important, and paramomycin, both microbial natural products, which are currently in the clinic for leishmaniasis. Maybe there are others that could be found. I don't know. It has been very, very challenging to find new drugs for leishmaniasis and indeed Chagas disease.
Thank you very much, sir. The rest are just praises for your presentation. Thank you very much, Dr. Paul, for your very our next talk is structure-based and AI-supported drug discovery targeting cortisol amino acid biosynthesis. And for that, I would kindly request Professor Dr. M.K. Paul on stage, please. Thank you.
All right.
If it works.
All right. First of all, thanks somewhat of Iqbal and Comstech for hosting us. As Paul said, we're really here to learn and to listen, and it's been a great experience. Everybody has been fantastically welcoming, and we really enjoy every day. But we also learn on really how these neglected tropic diseases affect the communities here, and I think that's really important for us. And we're really trying to make all our relationships as equitable as possible. There's one thing I wanted to add to what Paul said is, and just to give you an idea on sort of the amount of resources and money that has been spent on our diseases and other diseases. Our GCF network, Paul, correct me if I'm wrong, but we had 7.9 million pounds for this network. 7.8 million pounds, which sounds like a huge amount of money. But if you compare this with what pharmaceutical industries is putting into things like cancer or obesity or alcohol misuse, this is pitiful. I mean, this is just a fraction, percentages of what's been put into other drug design or other diseases. So we really have to sort of keep that in mind, that we are working on many extents on a shoestring budget compared to big pharmaceutical industries. But one thing what we're trying to do is using more than one approach. And Paul obviously mentioned sort of the parasitology approach, a lot of high-throughput screening, working with bugs and things like that. My background is a bit different. My background is structural biology, chemistry. And so we're trying to use the more structure-based approach in parallel with what is happening in the laboratory. And I think that's looking at the successes on drug development. That's one of the key issues. There is no one solution and not one single strategy which will give us a new drug or new therapy. So we really have to work in parallel. We have to work together in order to get some progress. And in this stage, I actually had a question for you, because I don't know sort of the background. So I just want to show of hands who would call themselves a biologist. The biology background. That's a lot, okay. Mostly biologists. Any chemists? Biochemistry comes next. Biochemistry, pure biochemistry, computers. Computers, very good. Any structural biology? Okay, so you're gonna be in for a treat then for structural biology. I'm gonna start a bit back on the structural biology. So I've been working on structure-based drug discovery or method development since my PhDs, and I think I just want to sort of remind you that really this notion that we actually use the exact three-dimensional structure as a starting point for drug design is really as old as any first protein structure determinations. I do hope that everybody's recognizing this as an alpha helix. Oh, louder. Okay. I'll try to shout. There you go. All right. So really the first structure-based drug design approach comes with the first protein structure that has been determined. And again, I would hope that all biochemists recognize this as hemoglobin, the second crystal structure that was determined. And right after the structure was published, people tried to find allosteric inhibitors that would sort of sit here in the interface to tackle sickle cell disease. So really the idea that I have a three-dimensional structure, defined structure, and then I use the structure to define molecules that sit here and do something is really as old as structural biology. Now, needless to say that these attempts were completely fruitless, and there's still no treatment from sickle cell disease. But it should also be said that's a particular difficult problem. And I come to sort of the difficult and more amenable problems a bit later. There you go. So one of the first really great success stories for what we call a ligand-based drug design approach. So we take a ligand-based, it's called captopril, which is a treatment for hypertension, which was developed in the 1980s. So this was the first big blockbuster drug, which was based on knowledge of the three-dimensional structure. In this case, it was really the structure of the ligand, not the structure of the protein. And it's actually based on a natural peptide that was discovered in Brazil, in the jungle. But the companies then basically made similar compounds, and this became one of the first $1 billion drug treatments for the company. So huge success. In terms of fast structural biology of proteins, the first big success and really breakthrough came in the 1990s when the HIV protease inhibitors came onto the market. So these are the first real treatment of HIV. And I actually remember this. I was a postdoc in Seattle at that time. There was a big conference up in Vancouver when this was announced. And it was really in the lab. There was sort of a buzz in the lab because we could see there's the first huge success of a really structure-based approach that not only made the companies hundreds of millions, but actually saves millions and millions of lives. And HIV protease inhibitors really converted HIV from a best sentence into a manageable disease. So it's a huge success, and it really sort of invigorated the field. We also at that time, so again, I was a postdoc in Seattle. We had the chief scientific officer of one of the big companies giving a big talk to us, all postdocs, all very ambitious. And he was basically sitting there and saying, you guys can never make a drug because we invested $700 million. That was back in 1996, $700 million, and you just have $10 million. So none of you will ever succeed, which was a bit disheartening. And I think he was wrong. But it was just to sort of give you an idea, the effort that was put into these blockbuster drugs. Obviously for HIV, this perfectly paid out. Now, on the next one, you probably recognize this as a three-dimensional structure. And I would ask my colleague Paul if he can tell me what the structure is. No. No. Anybody? Yes, it is the HIV protease. It's the HIV protease. And I show the structure really in contrast to all those complicated transmembrane proteins that Paul has been working on. This is a trivial structure. It's really simple. Just 100 residues up here, 100 residues up here. It's a homodimer. It's nicely resolved. It's easily makeable. I mean, a high school student could make that protein today and crystallize the protein. It crystallized right away. So in terms of structure-based drug design, it was a fantastic, ideal, simple target. Even easier, it was a protease. And back at that time, hundreds of protease inhibitors were already known. So you can basically go into a company and buy 10,000 protease inhibitors. So your starting point for structure-based drug design is incredibly easy. Unfortunately, most of the targets today become a bit more difficult. But I just want to give you an idea on how much more complicated many of the targets are that we are sort of trying to work with the sphingolipid biosynthesis. Now, as I said, the structure was one of the key features in order to develop these. So this is the same structure with one of the inhibitors. This is actually one of the drugs bound in the center. And again, another thing that we see is that the ligand itself doesn't change the structure of the protein significantly. That sort of comes a bit together. It goes a bit like this. There's no big structural changes. And again, in terms of structure-based drug design, that's a perfect case because sometimes these proteins move quite a lot. They're much more dynamic. And even the binding of the ligand or the binding of the substrate changes the structure of the protein. And that, of course, makes everything much more difficult. So again, HRV protease inhibitors, fantastic success story, really changed the field, but at the same time, sort of the easiest example that you can think of. And unfortunately, we have not found a simple target like that for any of our neglected tropical diseases. So, so we have been working on pathogenic trypanosoma, and I just don't need to repeat this too much because Paul has done this already, and I'm also not a biologist. So what I need to remember as a biologist, as a non-biologist, is that, well, they have different stages. So this is, again, our insect stage, and then they have a human stage. So they do encounter many different environments. So that's something I want to keep in mind. Right? The ones that we have been interested in, and just as a repeat, is trypanosome and leishmania, and in particular, trypanosome cruzi, up here, Chagas disease, and Leishmania species, including Leishmania infantum. And I'm mentioning Leishmania infantum because that's actually the enzyme that we have been working on. And I should point out that this beautiful illustration comes from my PhD student, Kate Saul, who has just finished her PhD. So thanks for that. Thanks, Kate, if you see me. There you go. So different environments are important, and this is important because we look into the amino acids in protozoa. And this is work based on Ariel Silber, who is part of our network in Brazil. So again, I'm a structural biologist, I'm not a microbiologist or parasitologist, so I need to rely on them to tell me what's important in terms of biology. So these pathogens encounter really wide-ranging conditions with very different availability of the 20 amino acids. So one of the questions that Ariel has been asking is, how does the pathogen adapt to these different conditions? And what he has identified is that they're basically amino acid transporters for most of, if not all of the 20 amino acids available in genome analysis. So in principle, it seems that the pathogen is able to scavenge amino acid as nutrients from the environment. But of course, these are transmembrane proteins, so they're complicated proteins, they're much more complicated proteins. So very few of these have actually been fully characterized. So the genomic data set tells us, yes, there's probably a gene out there that is responsible for getting this amino acid into the cell from the host, but really how it works and under what circumstances this is present is really not clear. Right? But in addition, and that's sort of curious and comes to, again, what Paul says, these pathogens are highly adapted and they have multiple systems. So not only do they have these amino acid transporters, they also have the amino acid metabolism pathways, which basically go up to glutamate as a central path, feeding into the TCA cycle. But these pathways may also allow the biosynthesis, the de novo biosynthesis. So in principle, the pathogens seem to be able to scavenge amino acid, but once they encounter conditions where the amino acids are limited, they can also make the amino acids de novo, right? So the ones that we have been concentrating on is actually cysteine. And the reason is this is an important part of the redox control during the infection. So the redox control is crucial because within the macrophage, the pathogen, and again, Paul, correct me if I'm wrong, has a particular strenuous redox condition. So basically, the pathogen needs to maintain its redox potential. And this has been done by a number of molecules. So this is mainly done by molecule trypanothionine, which is this one up here, and monoglutathione spermidine, which is this one up here. So these are molecules that can be oxidized and reduced, and therefore they can maintain the redox potential within the pathogen. Right? Now, why is this relevant for us? This is relevant for us because they both contain cysteine as a building block. And now I can ask a very quick question. We all recognize the cysteine in these molecules. I go to this side. Obviously, I go to the other side. If you look at these molecules, use a bit of chemistry, and you see the cysteines up here. There you go. Right. So cysteine is the central amino acid that is relevant to maintain the redox control of the pathogen. Again, this work by Ari Silva, and so he has looked into the cysteine de novo biosynthesis in a bit more detail. And in principle, there are two different routes for cysteine de novo biosynthesis. One starts with serine. So that's just a pure coincidence. That's the same amino acid that you mentioned, Paul, right? With serine acetyl transferase, which makes this O-acetyl serine, which then is taken up by the cysteine synthase, makes it into cysteine. And the second pathway comes from homocysteine up here, and it's catalyzed by these two proteins, cysteine B synthase and cysteine lyase. So it's basically two pathways that lead into cysteine. So again, this shows sort of the flexibility of the pathogen. So not only can it scavenge cysteines from the outside, it can also make cysteine from two different pathways, which in terms of a drug target doesn't really make it ideal. And we've been sort of some discussions on how important or how relevant these proteins are as drug targets. We come to this in a moment, but I think the key part is that based on preliminary genetic data, these two proteins, in particular the second one, cysteine synthase, have a potential as a drug target. So we clearly see an effect on genetic knockouts. But I should also say there's also practical considerations. All of these proteins are nice drug targets or potential drug targets in the sense that they're all soluble proteins, right? They're all cytosolic proteins. They're not membrane-bound proteins, and they're basically proteins that we can work with under limited circumstances. So basically, these are proteins we can work with as a PhD student. And I'm very clear, we are very clear, we would simply not risk an entire PhD project working on a membrane protein where somebody spends 4, 5, 6 years of trying to make the protein and nothing happens. So we have to be a bit pragmatic as well. And the cysteine synthase just proved to be actually a really nice, target, as I will show in a moment. And of course, working with a protein that can be made and we can do things with and develop assays allows us also to develop methods, which is one of the other parts that we were doing. So cysteine synthase, there we go. Too fast. So what we then did, go back, we looked at cysteine synthase targets, and we basically decided to look at the number of cysteine syntheses from closely related organisms. So we again looked at T. cruzi, Leishmanium elephantum, and we also added Trypanosome theileria to the mix as a third pathogen, which is actually the cattle pathogen. Now, you might ask, why would we look at these enzymes that are all fairly similar? I mean, in terms of sequence identities, this is just a sequence alignment. Without going into detail, sequence identities are about 70%, and all the residues of the active site are conserved down here. So for one, it really shows us, gives us an opportunity to investigate the differences between closely related enzymes. So these enzymes for, I would say, biologists look almost the same. But for biochemists, they're actually different, the differences. And maybe it's these subtle differences that are relevant. And we often learn something from these subtle differences which don't seem to be relevant for people who see a protein just as a blob. We do see proteins as molecules with single atoms, and really single atoms can make the difference between life and death. So I'm quite interested in that. But again, there's also a pragmatic reason. I'm a crystallographer, and as someone knows, without crystals, we are nothing, and no PhD student gets a PhD without a crystal. And having 3 different proteins simply increases our chances of getting these high-quality crystals that we need for every consecutive step. Right? So again, we went with these 3 enzymes, and I just go over a bit of the methods that we're using. And of course, this has recently been published. Again, Kate, my fantastic PhD student, who will be doing a postdoc in New York City in February, has done all of this work. So really, I would say straightforward, simple work. All our proteins are overexpressed in E. coli based on PET vector. So this is standard overexpression systems. In effect, because there's just a few of these genes, we simply buy these genes from the transcript. I have not cloned a gene in my life. I just bought the gene. They're cheap enough. Obviously, we can't do this with hundreds of genes, but with 3 genes, we can do this. And it just works really well. In this case, all of these are purified quite easily with standard chromatography methods, can be done in pretty much every molecular biology lab. First step is usually a HISTAG, nickel anti-A column. There's always a polishing step and so on. And again, I'm referring to summer. We know that crystallization requires the most pure samples. So we spend a lot of time in really trying to get the most pure samples. This is just one example. These proteins highly purified. There's really almost no contaminants out there. And spending this time on getting the protein sample as pure as possible pays out at every consecutive step. Right. We have extensive quality controls, and I'm just showing one picture again to please my colleague Paul. This is the activity assay. We did actually develop an activity assay in the 96-well plate, which can actually be used for later screening. So again, once you have enough protein available, it's easy to develop then the activity assay in a 96-well plate. So if you wanted to do some high throughput screening with more compounds, this can be easily done, right? The other techniques we use extensively is mass spec, circular dichroism to see that the protein is properly folded. And in particular, I'm a big fan of these thermal ship assays because they can be done very easily to find the best constituents, whatever the application is. And if anybody is working on protein chemistry and trying to make a protein stable and happy, please come to me and talk to me later. I would say by now, half of the work that we do in the lab is actually trying to make our protein happy, and keep it happy, rather than doing crystallization. Everything else can be done automated. But here, there's a lot of sort of fine-tuning and attention to detail. And, I mean, I really can't praise Kate enough because she was really excellent at that, which is proven by the fact that all 3 proteins crystallized. So once we have pure protein, we go to crystal structure determination. I won't be talking a lot about Brian. So basically, these starts with high throughput crystallization done in 96-well plates with robots. We typically set up 10 of these trays at 2,000 conditions. And then the student has to look at all of these 2,000 conditions. And although there are imaging systems available and there's image recognition software available, none beats the human brain. So finding conditions out of something that looks a bit like this and saying, okay, this is promising because these little blobs look a bit crystalline. No robot, no image recognition is as good as a human brain that says, promising, let's go on. This can take days or weeks. It also can take forever, particularly if we work on a membrane protein. As I said, that's just, it's why we have to have more membrane protein. So then we have to go to some optimization. This is usually done manually, optimizing your crystallization conditions. And again, this can take weeks to months, and we might get a crystal of this particular size. And then we might try to pick this crystal up. And this is sort of, again, a question for you. We think about crystals, we all recognize a crystal as beautiful diamond-shaped crystal. How big do you think a crystal needs to be? And I really want you to talk to your neighbor and think about how big a crystal should be. Talk to your neighbor, think of how big a crystal needs to be, a protein crystal, so that we can do something with it, and give me an answer. Right. Okay. Anybody want to give me an answer?
Diffraction,
How big? How big the crystal should be?
It should not, I think the diffraction factors must also be considered,
Yeah.
considered. It's a main factor, one of the main factors.
Yeah.
And the size also matters.
Good.
It should be not too much high, small size should be small.
So
That like if index should be, I don't, is there anything, but should be small.
Millimeters. I hear millimeters. Anybody else? Well, luckily we can go to things like 10 micrometers. Right. This is actually one of the crystals. This blob is about 20 micrometer. So 0.02 millimeter. And that's something that a lot of people don't realize. These crystals really can be quite small. I mean, an E. coli cell is a couple of micrometers. So these crystals are just a bit smaller than cells. Now, the only challenge is that when we do crystallography is that a lot of these things can be automated. The one thing that cannot be automated is picking up this crystal, right, in a little nylon loop. This is a little nylon loop that's about 100 micrometer. So under the microscope, I see some are smiling. Somebody has to pick up the crystal and put it into liquid nitrogen. Again, people have tried to build robots for that. The Japanese group has, they've made a little top string robot, and of course, what it does, it's the crystal. Wonderful videos on there that John, and people have given up on that. So we still need a human to pick up the crystal, put them into liquid nitrogen. And I just mention this because we're coming to the point that we do everything in high throughput. So again, that's the one thing that we can't do high throughput. So this mounting, I said 10 minutes per crystal if somebody is experienced. If you see somebody doing this for the first time, it could take a day. So anyway, we then ship the crystal to the synchrotron. We normally ship it to the diamond light source, and but other synchrotrons are of course available. And then due to the advancement of these synchrotrons, these high intensity X-rays, the actual data collection takes a minute per crystal. We can collect thousands of crystals. And again, just to put this into perspective, when I was a postdoc in Seattle and I went to my first synchrotron trip to Stanford, we collected a data set for 12 days on a single crystal. So a single crystal was on the machine for 12 days without interruption. The same data set would have been collected today in a minute. It's just the advantage in synchrotrons is just absolutely staggering, right? And then, of course, computers come into place. The structural solution itself, if it's a simple structure, takes minutes, right? And it takes a bit more refinement to make it publishable. But these points are really, really fast. So crystallography has come to a point that it has become a high throughput technique. I'm just showing the different crystal structures to show off and to show a collaboration with our partners in India. So this is a structure that we actually solved for somebody in for Ashish Aurora in Lucknow. And what they have actually done, they sent us the crystals. So they crystallized the crystals in Lucknow. They sent us the crystals to Durham. We sent them to the synchrotron. We collected the data set. We sent the data set back to India, and that they did all of the work. So it's a really nice example of cross collaboration within our network. I'm not going to talk about the structure itself. So again, Kate managed to crystallize all three targets, which is brilliant. So this is the TCCS structure at 1.8 angstrom resolution, and I see some people have an understanding of resolution, so that's pretty good resolution. We're happy with that. Leishmanian and Phantom 1.75 resolution, and the runs of Thalera a bit lower, 2.7.5 angstrom resolution. So these are all very highly resolved structures, and you can probably see that they all pretty much overall have the same structure. And again, we would totally expect the sequence identities is about 70%, right? So if you just overlay these structures, the overall fold and the active sites are highly conserved. And some people might ask, okay, why are you bothering to solve the structures where they're similar? Well, first of all, we want to prove that they're similar. But this is actually really good news for future drug design, because obviously we could then see that if anything binds to the active site, it might actually bind and inhibit all the three different enzymes, and might be good starting points for more selective inhibitors. So in that respect, I'm quite happy with that. But then we always see some differences, and we actually saw some loop flexibilities, which gives us a hint of protein dynamics. And I come to that later. So hold your thought. The structure is one thing, it's great. The proteins also wiggle. And the wiggling of proteins is important, and we might want to consider this later. But also this is protein dimer. So there are some unexpected changes in the protein in dimer interface. And again, that's something that is really difficult to ascertain a priori. I mean, we could see, yeah, the structures look overall the same, but then again, in the interfaces, there might be some significant differences, which we might be able to exploit. So I would say Any protein that crystallizes is important, and always learn something unexpected, even if the structure itself looks quite similar in the first instance. Right. So if you look at the active sites, so one of the things that we just happened to notice is that the three different structures show different states in the reaction cycle. This is nothing that we planned. We didn't try to track any intermediates, but because all the conditions, the crystallization conditions are different, we might actually find differences in the active site, and that's what happened. So our TCCI structure has the BFP, that's our cofactor, covalently bound, but it also has the substrate or acetylserine in the active site. So basically, it's this side up here. So we find our PRP up here. It's covalently bound in as a shift base to the lysine, and we can actually see the electron density here. This is the unbiased electron density, so we see it's continuous to the lysine down here, and I've shown the OS that sits up here. So this is one state within the reaction cycle, right? And for reasons we don't know, it's just happened, coincidence. The Valeria CS has only PLP covalently bound. So again, it's nicely bound. It's not quite as high resolution, but it's nicely bound. Again, covalently bound. And the Leishmanium infantum is actually empty. So this is an empty site. There's no PLP bound site. So it's a site before the PLP is covalently bound. I would say this was a bit of a surprise because the protein clearly was yellow. So we expected the PLP be bound, but it's just the way it is. We're not going to go over the cycle itself, but basically out of the different cycles, we found different states within the active site. We can basically see where the dynamic changes happen. One of the things that we can see is the active site loop that moves over the active site when PLP is bound. So although the overall structures of all of these three crystal structures look really the same, the three differences, the local differences that tell us something about what's happening during the catalytic cycle. But we're talking about drug design, not catalytic cycle, so let's move on. Yeah. So and then again, I always point this out. Whenever we solve a crystal structure, we find something. I always find something interesting. I haven't come across a structure that I didn't find interesting. It's always something that, oh, that's neat, right? And this then appears TCCS crystallized with two independent dimers in the asymmetric unit. That basically means that we have four molecules that are independent in our crystal structure. And for the summer was an interesting pseudo-symmetry with beta angle being 19.1 degrees. So it was the crystallographic challenge, which was very nice to solve for K to learn a bit of crystallography. Why is this functionally important? Well, and this is functionally important because it also gives us an idea about dynamics, because it really shows, again, which part of the molecules can move and which part of the terminal can move, because there are different conformations, and which parts of the molecules are really static. They are really identical within these four different molecules. So again, we learned something about dynamics from these different molecules in the asymmetric unit. Right? And the second thing we found is a bit of a surprise. We did find a glycerol molecule and a ribose molecule in the hydrophilic pocket in the interface. So this is the dimer interface. In one case, we see something like a glycerol molecule. It's found here again, shown with the experimental electron density. And the other dimer, we do actually find a ribose molecule, which probably comes just sort of sits there bound from the purification. Now, on the one hand, these are perfectly artifacts of our treatment. So the glycerol molecule is probably rather coming from the crystallization itself. We often have glycerol in our crystallization because glycerol often just makes proteins more stable. Glycerol is one of these magical molecules that just stabilize proteins. And one reason it stabilizes, it just occupies this pocket, right? And that probably helps it to just keep it a bit more together. And again, pure coincidentally, the second one is a bit of a larger blob, and this clearly, in terms of hydrogen bonding pattern, is a ribose molecule. And again, I don't think that has any biological relevance, but it does show us a pocket which we wouldn't know without our crystal structures. And of course, protein-protein interaction inhibitors is one of the hot topics in drug design. So what we basically have identified, we have identified a hydrophobic pocket within our dimer interface, which could be a starting point for designing a protein-protein interaction inhibitor. So if we had anything that binds into this pocket and interrupts the interface so that the dimer breaks apart, the protein can no longer function. So again, it's just one of those coincidences that we see in a crystal structure, and we might be able to exploit a little bit later. Right. But obviously, well, the structure is one thing. We're really trying to go to drug discovery, and the method that we use is fragment screening. And fragment screening really has become sort of a standard method in academia, but also in pharmaceutical industries. We work in terms of our PhD program with a number of companies, and every single company I know of, they have their fragment-based screening protocol, where they use fragment screening for drug discovery. So not screening entire large libraries, but screening relatively small number of compounds, maybe 100 compounds, which are fragments and starting points for then drug discovery. And it's easy to do, and it doesn't require huge amounts, so we can do this as well. So in this case, we choose to focus on the TCCS and LSCS, not because they are the most important, but because the crystals were simply better. We had hundreds of crystals, we could make a lot of crystals, and they were available. And we also started with the fragment screening library, which is called FragLite, which was developed in Newcastle by our friends and colleagues in Newcastle. So the reason with working with FragLite was simply we could get the compounds for free, and they work well. That's the compound library of about 31 compounds. So all we have to do, because then, okay, we grow crystals, hundreds of crystals, right? We then take these crystals out, and again, this has to be done manually. Somebody has to very carefully take the crystal out, put them into a new drop, and then soak compounds into the crystals. So we leave them there for like 10 minutes, half an hour. And then we go and send them through the synchrotron, do the X-ray diffraction experiment, quickly solve our structure and see if our compounds is bound. And this actually worked really well. So I'm not going to show you all of the fragments because that would be repetitive, but I'll show you one of them. It's not the best one, just the first one. It's called FragLite 18, shown here. And so again, the blue density is our experimental density. This is where the fragment is bound. It's already indicated as a nice hydrogen bond up here. But these FragLites, in particular, they do have a bromine atom. And the bromine atom can be used to identify our fragment and really position our fragment, even if the resolution is not very good. Basically, our block, that is our ligand, is not very well defined. We know from experts the anomalous scattering exactly where our bromine atom is. So the green density, again, for the expert is our anomalous difference map. So the fragment is identified. So this really gives us a starting point on how to make the ligand, starting from this little fragment. And the idea, of course, behind this is that in terms of binding energy, the majority of binding energy comes from hotspots of binding. So this is a hotspot of the active site. So all we would need to do is just use a bit of chemistry and make a larger compound. So we've got 6 crystal structures with TCCS, and we have 8 crystal structures with LCCS. And if we superimpose all of the ligands, right, we can really then identify the different binding sites. So again, it's not just about finding one single binding site, it's getting an idea about all possible binding sites on the protein and really trying to figure out which one is the most likely to work for any structure-based practice approach. So this is then a blow-up of the same picture. And the first thing you probably notice right in the middle, there's again our dimer interface. So the fragment screening identified the same ligand, the same hydrophobic pocket in our that we have already seen the ribose and the glycerol. So it's a really nice confirmation that this really exists. It's not just an artifact of one particular crystal structure. No, it really exists as a nice pocket that we could use if we wanted to go on and try to make a protein-protein interaction inhibitor. I should say we didn't go for this in the first place because protein-protein interaction inhibitors are notoriously difficult to make. So we choose something more easy to start with, right? We also found some in the active site. So this is down here, the active site down here. But then we actually find a second binding site. Well, we call this an allosteric binding site because it sounded better. We're not sure if this is really an allosteric binding site, but there's certainly a second binding site. This is the same up here, these two binding sites, right? There's certainly a binding site where a lot of the fragments bind, right? So with the fragment screening, we identified a second binding site, which we're going to explore in more in the future. And the nice thing about the second binding site, this is probably more specific compared to our active binding site. If you look into the active binding site, this will look very similar in every cysteine synthase, but will look very different in many PAP-depending proteins, right? This down here is something that is very likely to be much more specific to our specific cysteine synthase from these pathogens. So in terms of priority, we actually think this is the most likely highest priority binding pocket that we would explore. There's also a couple of binding pockets sort of at the edge of the molecule, and these are probably crystal complex. So if we think about a crystal, we would have the next molecule down here and the molecule down here and so on. So these just sit between molecules that are packed together in our crystal. And those are not very valuable. So my point is, it really needs, again, it's a human brain. It's a human, Kate, not me, to look at this and analyze all of these binding sites to decide really where to continue. Right. Breathe. I think Paul is telling me speak slow. Right? Good. So from this, I'm going to switch here a bit and talk about AI and structure-based drug design, because I'm going to show how we use the AI, but also sort of bring this in where I think AI is and where the current state is. And I hope I spark some discussions. So particularly people with a computer science background might disagree with what I'm going to say. So in principle, AI is being used everywhere at the moment. I mean, you find an AI application and everything. If you Google AI, chemical synthesis, new biological target, right? But we really want to focus here on the relevant steps in structure-based drug design. So the relevant steps is 3D protein structure prediction, protein ligand interaction predictions, drug activity predictions, and de novo drug design. If you type in de novo drug design in Google with AI, you find, I don't know, 10,000 hits. So I'm going to give you a few hints about what I think about it. Now, obviously, the power of protein structure prediction. We all have available are aware that AlphaFold and RosettaFold have revolutionized structure biology. So this is just the interface of the AlphaFold database, which currently contains 200 million protein structures predicted. 200 million. So there's a dataverse, a data universe that is far too large to grasp by any human being. Right, so it's an absolutely amazing resource. And when this came out, I did get about half a dozen emails telling me that I'm unemployed now, which is not true. Now, I should say we could run our CS structures through AlphaFold, and the structures itself will be reasonably well predicted. In fact, they will look pretty much like the structures that we have seen. So in terms of overall structures, AlphaFold would solve the structures as well. And just give you a hint, AlphaFold is actually very good for novel structures as long as they're not too large. And if you're interested, this is the structure we saw before AlphaFold came out, which is a really unusual protein in terms of sequence. And when we published the paper, the referee said, "Oh, what did AlphaFold do to this?" And so we superimposed the structures of AlphaFold and RosettaFold. This is red and teal. Teal is our structure, and yellow. And sort of the overall structure is actually quite nice. Right, so AlphaFold could have been used to solve our structure with molecule placement. But I think what really is important, and again, if you want an example, read this paper, really doesn't tell you anything about the biological function. It is still, and that's what we're interested in. We're interested in biological function. We want to use the structure. So AlphaFold, great tool for structure prediction, but somewhat limited. And I'll give you more examples of that. So in one respect, AlphaFold really doesn't tell us much about the dynamics, right? And we know that protein dynamics plays an important role in the function of proteins. And I've just sort of this cartoon of a molecule that sort of wiggles around, but it's actually this wiggling around that is absolutely crucial for this particular protein to function. And if you're interested, I can explain why this is for this particular protein. And AlphaFold really doesn't know anything about dynamics. can't tell us which parts are moving and why which parts are moving under which biological function. So it's one of the huge drawbacks. I know, and you will be seeing papers coming out in important journals that claim that AlphaFold tells us something about dynamics. I'm happy to dispute that with when this paper comes out. I've only seen the preprint. Anyway, and then of course the proteins often have unstructured parts. So this is my favorite AlphaFold prediction from a protein that for some reason I was interested in is human was it SET binding protein 1. So this is the 1,000 residue protein, and that's a prediction that AlphaFold gives you. And do I have 30 seconds to let them ponder on the structure? What do you think?
Yeah.
What do you think about this? Are you happy with that?
Yeah.
No. No, why not?
For that, so there conveys much confusion regarding active sites and also the allosteric binding sites are also confusing. There is no, I think the active sites is not predictable.
You're far too nice. This is just utter nonsense. And I mean, this is just oh, hang on, 1,000 residues of unstructured protein, and I really don't know if the human makes a protein of 1,000 residues that's completely unstructured. I mean, just can't see how this functions. Right? Now, what I don't know is, well, does AlphaFold not find the structure because the sequence of this protein is so unique and there's not enough sequence information, right? AI relies on the data that you put in. If there's no information about the sequences here, well, just can't, right? But we don't know that. It could also be that, yes, I'm wrong, and this structure really is completely unstructured, and that's what the function of the proteins, and we really don't know that. As I said, this is just one of my favorite sort of nonsense predictions, but there are many, many of these predictions like that in the database. So just using AlphaFold as a predictive tool is not really useful. We did use AlphaFold on your proteins, Paul, and AlphaFold was very good in predicting the transmembrane helices. Perfect, right? But then again, we know there are 7 transmembrane helices in their parallel. I mean, I don't need AlphaFold for that. I can draw them on a piece of paper, right? My bottom line is you have to be very, very careful when using structure predictions. And it's still, and I have to use this term, it's the natural intelligence that is needed to guide the artificial intelligence. There you go. So AI and protein ligand interactions. So this is sort of the next time when we come to protein ligand interactions. Ideally, we would like to calculate binding energies a priori. So we have a protein structure, we have a ligand structure, we should be able to calculate the binding energy, right? And there's really various papers out there that describe applications to AI predicts. Probably most well known is Delta Vina, which is part of now Autodoc and Vina, which is a widely used program. It's publicly available. We have used Autodoc Vina a bit. We haven't used Delta Vina yet. There's another one called Potential Net, which seems to do quite well. But I really fail that there's a clear evidence that the results are superior. They're clearly faster, right? Because once the machine learning algorithms have learned their algorithms, the application is really fast. So the learning part takes a lot of computational power, but then the algorithms are very fast. But it's really questionable if the results at the end are superior. And I would sort of contrast this with what we call conventional methods, molecular dynamics simulations. And there's really an entire zoo of now very sophisticated computationally intensive methods that calculate binding energies quite well. Computers getting faster and faster. And I'm not sure if these will be fast enough so that these methods can actually compete. I would also point to anybody working on molecular dynamics simulations, look into MD, QM methods. I was just at a conference in Munich where some company people basically gave a talk on this, and they said, we don't do any MD anymore. We only do MD, QM methods because they're now fast enough. And again, in principle, quantum mechanics, well, solving the Schrödinger equation should be able to solve it completely. So again, questionable if AI really is going to make a difference in that. So in that respect, we did actually test one of those with AI. So what we did is, and again, it's based on the collaboration with friends from up from Newcastle and Nicole. They developed this program, FE-Grow, for growing our fragments into ligands. So we basically said, oh, we've got all these crystal structures. Why don't you use your program and again have a look at that paper? It's really well written, very well described. It's all open access. It's good software. And we said, we give you our data, give us a new ligand. So let's see what you come out with. I'm not going to go too much into detail, but all I'm going to say is that the FE part, so the AI part, is actually only in here in the binding scope. So this is quite conventional. It takes the ligand and then it uses basically makes additional molecules based on your fragment. So it makes fantasy chemistry, so not based on synthetic chemistry. And then it calculates binding energies based on a mixed molecular mechanics and machine learning function. So the machine learning is in the calculation of binding energies. So basically using part of what I've just said before, where we replace the phosphates, or we make the phosphates. So it's really fast. That's the advantage. All right. So they came back and said, great. A couple of weeks later, gave us a compounds, 150 compounds, and we can look at some of them. So the advantage is it's really fast. Fantastic, right? And the other advantage is it explores a vast theoretical chemical space. So the chemistry that they give us is not restrained by what is synthetically possible, which maybe an organic chemist might not see as an advantage. Right? The disadvantages, again, so the protein is really considered as non-flexible. This is still a very simple molecular mechanics force field enhanced by AI, but the protein stays unflexible. And of course, we know that when the ligand binds, that at least the side chains move. There's always some little movements, right? So we see an example here where sort of the ligand sort of goes into the protein surface. The other thing that we noticed, and Paul alluded on that, is it doesn't know anything about medicinal chemistry. So we found quite a few compounds that have CN and nitro groups, really things that medicinal chemists probably wouldn't touch with a barge pole. So on the one hand, yes, it's not limited or restrained by chemical synthesis, but at the same time, it also doesn't know anything about medicinal chemistry. And I would consider this a disadvantage, right? It also lacks any considerable consideration. So we found compounds with 3-membered rings. And I mean, I'm a chemist by training. I would normally not try to make 3-membered rings. They're usually difficult. And also chiral compounds, well, maybe one chiral center, but as soon as I get a second chiral center, things just get too complicated. So again, advantage, it's not limited by organic synthesis. Disadvantage, it doesn't consider synthesis. So we fed this back to the guys, and they said, okay, we can do better. And one thing they can do is they use the enemy database. So enemy is a company they're actually located in Kiev, and they have developed a synthetic sort of combinatorial synthetic method where you can download a database of, I can't remember, a billion compounds, right? And basically, they can make each of these billion compounds. So great, it's really fast. So they ran exactly the same through the enemy database. And this is one of the compounds they came up with. And basically, we can now go to enemy and ask them to make the compounds for a few, I don't know, $50 or something. So it's not too expensive. So it's possible for a limited number of compounds that we definitely going to do this. So I would say there's certainly some space. All right. So the other thing I had is AI and drug activity predictions. And again, plenty of stuff out there, right? And it seems that, so just from the literature, machine learning techniques, they seem to be very good at predicting toxicity. There's a sort of a good paper, maybe 8 years ago, from the Tox21 data challenge, where they basically gave 12 tox data points for 12,000 compounds as a learning data sets to different companies. And then as a competition, who did best on 600 unknown compounds. And again, they did quite well. And I would expect them to do quite well, because this is a very good and very accurate data set. So one of the problems, of course, is that it's always based on the data set. This is a good data set, you have 12 points, 12,000 compounds, large learning data set. But I've also spoken to experts, and well, it's really hotly debated if the methods are superior to any of the given methods. And obviously, when you write a paper at the moment and you try to get AI in the title, your impact factor rises, right? So everybody's trying to sell it as AI, and try to show that AI is slightly better than conventional methods. But I would say it is hardly debated, and I don't know the answer to that. In any case, tox data needs to be experimentally validated. So in that respect, if a method is as good as other methods, but I still need to do the experiment, why would I waste my time on using this if I have to test it anyway? I don't know. All right, I'm coming to this. But then there are difficult cases, and I think Paul has elucidated to these really difficult cases that in many cases drug targets are poorly defined. I don't think in any of the drugs for neglected tropic disease there is a defined drug target like for HIV protease inhibitors. They might not be accessible, right? They're simply not. We can't make the proteins in any reasonable amount, so we can't do any high throughput screening. Many cases there are multiple. This is even worse. So we don't know which one is the most important. And in fact, the most important might not be the same for each species or at each life stage. And I think for about half of our drugs on the market, we don't know. We don't even know what the outcome is. So this is where I think AI actually does have a role. So if we really see this as a pure black box, we have an input layer of data. This could be our crystal structure, chemical structure, or physical data. It's really hard data, right? And if you look at this, this is hard data that basically has no error. We know what the molecular weight is. We know the three-dimensional structure of our ligand really well, right? And then we have soft data. So we might have high throughput binding data. We're lucky. We might have some data on cellular assays. This is data, soft data in the sense that this has got an experimental error, but at least it's accessible. And we can weigh this data according to its experimental error. And that's a really important part in terms of machine learning and statistics, that we weigh the error according to how reliable the data is, right? And then on the outside, we might have something like our output layer, which is any biological data. I've stolen this from one of my other presentations where we work on neuroregenerative disease. So this could be neurodegeneration. It could be something like stress, proteomics, behavior in animals, whatever. Right? Again, it's quite soft data, but again, this is data that might be available for our compound. But again, we don't know what the target is, and this is where sort of a neural network like this might actually work, where we really have a learning data set, right, that decides which of these neural pathways are relevant without really knowing the underlying biology or the underlying chemistry, and then come to an output layer that actually gives us some predictive measure. And there is actually a huge, and again, anyone is interested in, there's a huge public-private partnership from the NIH, the National Institute of Health, where they've developed an entire framework called Atom AI or Atom Science, which is again freely available, where they're trying to use or include these models in different machine learning techniques into drug discovery. And I think I actually think there's actually a lot of lack of that. AI and the node design. I have to say I kept this empty because I couldn't find an example. I could not find an example of AI that really made the de novo design from scratch for any drugs. So I skipped this very quickly. Coming to an end. Two more slides now. Good. So I think just a word of caution. AI is in the news every day, right? And the reason is that people don't understand it, but if they use AI in their journalist article or in science, they got on top of the news. But really, it's so many different tools, right? That anything can be called AI at the moment, and it really depends on sort of which tools might be relevant for which application. So machine learning, deep learning, artificial neural networks, these are all slightly different things. And they will find their way in drug discovery and their niche where they are useful. But at the moment, there's also a lot of FOMO, fear of missing out, in particular from the investor world. Everybody thinks they have to be involved in AI because everybody else gets involved in AI. And I think, again, just a word of caution. And the biggest problem for us in terms of drug discovery, it really relies on the data sets. And if we can compare this with image recognition, and language recognition, the data set that Google has is vast. I mean, there are gazillions of pictures they can use for training their neural networks. And they have the entire library of the world to train their language recognition, right? We work with tiny data sets. We have, I mean, 10 compounds, 100 compounds, right? We have data on, I don't know, 10,000 compounds, right? So our data sets are much weaker. And if we look at the data sets that are published in the literature, unfortunately, not all of the binding data in the literature is accurate. Not even every single crystal structure is accurate. And often we don't know which data is accurate and how do we weight the data. So this is where the error-weighted merging of statistics comes into place. So I think the biggest problem is here that our data sets are really not very good. But as I said, nevertheless, I am convinced, and I think everybody is convinced that AI will find its niche in certain areas in drug discovery. And companies like Accenture are really trying to find exactly the point where AI makes a difference and doesn't replace natural intelligence, but really accomplishes and enhances natural intelligence. So in that respect, I absolutely believe that AI will change how we do drug discovery, but it's not going to be the AI press the button, here's a new drug for neglected tropical diseases, and not in my lifetime. So I think our jobs are safe. Right? All right. I just have this picture. I mean, everybody, in particular, the people who worked on this project. So Steffi and Kate are Kate is the PhD student, Steffi is the senior postdoc who did most of the work on the crystallography side, the entire team, all the collaborators. And I want to highlight that I want to thank the Biochemical Society because they were really generous. They actually gave me a bit of money to support this trip to Karachi and then the trip to Karachi and to Islamabad. So if anybody from the Biochemical Society is listening, thank you very much. It's really highly appreciated. And thank you guys for your attention.
Thank you very much, Dr. Paul. If anybody from the audience has any questions, you may ask now, please.
So, hi, doctor. So I have a question, like how far are we as a scientific community taking these like AI-based discover drugs to the clinical trials?
I think nowhere close. I think nowhere close. We're really just at the beginning. We're really just at the beginning. And I think what we need, actually, people like Paul and I, is the young people who really know AI in and out. I mean, I have a bit of a computational background, so I would say I have a vague understanding, but I'm not the person who's going to program things. Right? I've got one PhD student who might be doing that, and I'm very grateful. So what we really need is the expertise from the young people who know the ins and outs of the techniques. And I think we're at the beginning, and I absolutely it's going to change what we do, but not in the way that people think. It's not going to be the sort of the one solution. But certainly, if I was now starting my PhD, I would be looking for people who did Fortran. I did Fortran programming when I was a PhD student, ancient computer language. I would be looking into certainly AI programming, and particularly these large frameworks. I mean, there's a number of frameworks, and I would look into those that are applied by companies. So there's a 9 data. I mean, there's quite a wide range, but there are things that are clearly used by companies, and I would look into those so that I'm prepared to get a job at a company as well as in academia. That's what I would do.
Thank you, sir. Any other questions, please?
So considering the disadvantages of AI and how far its consequences can go, should we still rely on using AI in the discovery of medicine?
I don't see a danger in AI using in terms of developing medicine because we still need the entire experimental validation as well. So I don't see sort of a danger that somebody comes with an AI drug that comes into the market without the entire drug approval testing and so on. So I really don't see any danger. I see this as an additional tool that we should learn to use. Right? And it's different when it comes to language and ChatGPT and those things. And it's a very different area.
We have a question from one of the online participants here. So I think structure-based drug design is an important field in chemical biology. However, what in your opinion could be the best criteria for a protein-based target selection?
I mean, I think Paul could answer the question. We've been talking, we've been thinking about this a lot. The best target is a target that is validated genetically and chemically. So that would be the ideal target, is a protein that is really clearly identified both in genetics and in chemical test. I think the host homology for CS is not relevant in this case because humans shouldn't have system synthase. But I also should very clearly say that I have to admit, system synthase is unlikely to be a target of a new drug. It's a wonderful example where we could develop techniques and we could solve structures and work on methods. But I must admit, it's probably not going to be a drug target. Having said that, once we have a compound that inhibits system synthase in the lab, we will send this compound to Adila Silva in Brazil, and he promised us to test the compounds in mouse models. And well, we might be lucky.
Any other questions, please?
What makes bromine atom favorable to orient the fragment? Why not some other atom?
Say it again.
What makes bromine atom favorable to orient the fragment?
Okay, so we didn't use atomI, we used FE-grow. And really, the very prosaic reason is that Danny Cole has developed this up in Newcastle. We did actually also use a conventional docking. And in fact, what we did is we gave Danny for the FE-grow, the AI assisted, one of the binding sites. And we looked into the other binding site, the active site, actually with conventional docking. And we have a set of compounds that is based on conventional methods. And we're sort of playing them actually against each other, see who wins. The answer, and I think this comes from crystallography. When you work on crystallography, you're sort of used to work multidimensional. So it's always to do both methods. Whenever a PhD student comes to me in my office and says, should I use A or B? I say you should do A, B, C, and then you should come up with D, right? Do all of that. It's as simple as that.
Thank you very much. Chief, yes, sir, please.
I remember that we were actually discussing that the real crisis in pharmaceutical research is not the drug candidate, but probably the drug targets, because we know very little about complex biological systems. But if we get something which is phenotypically active and very active, is it possible to go into reverse pharmacology and try to identify the target with that? How would you do that if you come across such some compounds?
It depends on the set of compounds. So I would say there's a set of targets of compounds where I already know that it's not a single target, it's a multiple target, and it might even target things like the membrane. At that time, I would simply accept it, and I might actually go to what I had on my last slide, AI methods to really just use it as a black box where I have biological data, and physical chemical data, and try to link this again with a neural network without really knowing what's happening inside. If there's a clear idea that there is a target, I would certainly use techniques like thermal proteomic profiling. That seems to be the latest and most powerful method to really look into the binding sites. And the reason is that thermal proteomic profiling does not only look into the identify the proteins that the ligand binds in cellular, but also looks at the cascade. So, for example, if you see that a protein binds a kinase in thermal proteomic profiling, you will see the pathway of the kinases. So, which of the following kinases or targets are being affected? So, that's a very powerful tool. So, I think that's something I would do. Lots of work on probes, chemical probes. I would go to my colleague Patrick and say, can you help us chemical probes? I'm not an expert on that, but definitely.
You see, despite the fact that there are a lot of technologies available and not a lot being done in developed world, but in the from the developing world, if you still look into the literature, large proportion of the work is largely phenotypic.
Yeah.
And if they report something, I think there is some mechanism of looking into how this molecule or compound or set of molecules are actually working. So this is the reason I was asking.
I think, I mean, again, in our field, the lack of targets is the biggest problem. Absolutely. And unfortunately, I can't help on structural biology view. There is an idea that, of course, you can look at all the structures from the proteome, the predicted structures, and then based on the predicted structures, predict something that might be a drug target. I'm very skeptical with that, to be honest. Either we know what the protein is and the structure is there, and then the predicted structure is very good, but then it tells us, well, it's just another kinase, right?
Thank you.
Thank you very much, sir. Thank you so much. In the end, I would like to express my heartfelt gratitude. Question.
The hand raised, so
So he has already, yeah, we've already taken his question.
the hand is still raised.
Thank you, Dr. Hassan, for your question. I would like to express my heartfelt gratitude to Dr. N.K. Paul and Dr. Paul Denny for coming all the way from United Kingdom, gracing us with your presence and for giving very insightful, very informative talks. Thank you very much. Now I would like to invite Coordinator General Comstech, Dr. Iqbal Chaudhry, for his closing remarks and to kindly present the shields and certificates to the speaker.
I think I'll just spare the closing remarks.
Okay, thank you. Okay.
I've been just closing and opening, and I should do something else also. So there's some shield there.
Great.
There are, last 1 week. This is the only thing I'm doing it. But the shield which we are presenting is very confident. Yes. Yes, sir? You can see. Okay, so. Yeah, I'll tell you the story. This is a rare walnut tree. Endemic to only our region, okay? And the wood is almost eternal. Unless you really throw it in the fire, it will last. And so this is made of walnut wood, and there is a science here. Okay, so about comstech and about this particular tree and its benefits, its nutritional value, its pharmaceutical importance. And then it is comstech. When carved. Okay.
It's amazing.
So. Thank you very much for that.
Thank you.
And please keep going. I'm sure you would have probably a million of such stuff. You, sir?
Can I just ask, when did you carve that? When did you carve that?
I didn't know. I'm still learning. Maybe the next shield is in the.
Thanks so much.
But I think you have a good photo.
Thank you so much.
So we really plan to have a good photo. I think if we can go out and have the group photo in front of it, it'll take about 5 minutes if you all move forward. Shall we do it? So we'll go into the ground floor. You go out from this auditorium gate and I'll bring the guests with me.
Then please come back on this floor for lunch and your certificates.
Don't really go away.
Bring them back.